This video is about the role of isoniazid as a cure for tuberculosis. Mycobacteria are a genus of bacteria of which some species are able to cause tuberculosis. In this video, we will focus on the proliferation of mycobacteria, specifically the cell wall synthesis. Mycobacteria are distinguished in large part by the unique structure of their cell envelopes, the inner layer of which is simply formed by a phospholipid bilayer, of which the glycolipids extend into the periplasmic space. Cross-linked polymers of peptidoglycan provide shape, rigidity, and trigger counteraction, and highly branched arabinogalactan and mycolic acids form the core cell wall. Specifically, this viscid layer of mycolic acids encasing the exterior of the cell is what sets TB apart. This outer layer acts as a barrier to lysozymes produced by phagocytes of the host immune system, while simultaneously contributing to the integrity of the cell wall. Mycolic acid biosynthesis involves two distinct pathways, fatty acid synthase types 1 and 2, and 5 main steps, all of which transpire in the cytoplasm. In the first steps, Phase1 synthesizes long-chain acyl-CoAs with bimodal distribution between carbon-18 and carbon-26. Fatty acids by the Mannol enzyme A pathway, the latter of which undergoes carboxylation providing the alpha-alkyl branch of the mycolic acids. Elongation of carbon-18 by 2 carbon units by Phase1 yields the carbon-20 fatty acid, which is the starting point to the Phase2 pathway. Here it undergoes numerous rounds of keto reduction, dehydration, and anoxyl reduction. The product is a saturated long-chain myromycyclate of carbon-56. This myromycyclate chain is then activated and linked to the alpha-alkyl-CoA ester to generate alpha-alkyl beta-keto-mycolic acid. This process is catalyzed by PKS13. These mycolates are then transported to either the cell envelope or for attachment to the arabinogalactan present in the cell wall structure. The latter is done by the antigen 85 complex. Isoniazid is a prodrug that is responsible for inhibiting the formation of the mycobacterial cell wall. INH enters the bacterial cell wall via passive diffusion, is then activated by the bacterial enzyme CatG, which can be seen here. Phase 2 will synthesize mycolic acid via NAD+. The two will then bind, forming a reactive INH-derived species, which then couples with NADH oxidase, producing a nicotinyl NAD adduct. This will then bind to enyl-acyl carrier protein reductase NHA, responsible for the annulled reduction in the second step of the mycolic acid synthesis. This will then inhibit the synthesis of mycolic acid, which is essential for the integrity of the cell wall and for cell proliferation of tuberculosis. Antibiotic resistance is one of the biggest threats against the usage of isoniazid against tuberculosis. Resistance against isoniazid specifically is characterized by a mutation in the CatG gene. This very specific substitution results in a significantly reduced affinity for INH, which results in a great decrease at the rate at which INH-NAD adducts can form. Thank you for watching.